Good afternoon. So we're moving into the close of the meeting now, and it's nice to see that there are still a fair number of people here. Our next speaker is Sir Patrick Vallance, and he's our closing plenary speaker. But I worked with Patrick at St George's Hospital. We trained in clinical pharmacology as lecturers together. Our paths diverged thereafter. He went on to be chair of medicine and led the division of medicine at UCL, and later was headhunted to be Global President for R&D at GSK, and moved on to become Chief Scientific Advisor for COP26, and then on to be Government Chief Scientific Advisor from 2018 to 2023. He currently works as chair of the Natural History Museum, but I'm sure he does lots of other things as well. He had the pleasure of working through COVID and leading our response as a country to COVID. I'm not sure how much of a pleasure it was. We're going to hear a little bit about it, I'm sure. But he has the misfortune of being a witness at our COVID inquiry that the government's undertaking at this moment. And so he couldn't leave London to be here. So he's recorded his presentation for us today. One thing I will say is I think his talk is about 45 minutes and he's not able to do the Q&A. So we're going to, you'll hate me for this, but we're going to move everything forward 15 minutes and finish slightly early. So I'll hand over to Patrick to deliver his talk, and then we'll have the closing ceremony to follow.
Hello everybody, my name is Patrick Vallance. I, until recently, was the UK Government Chief Scientific Advisor, and I am by heart a pharmacologist and clinical pharmacologist. And I'm incredibly sorry that I can't be with you in Glasgow. I was looking forward to catching up with colleagues and friends from pharmacology and clinical pharmacology. But what I hope to do is give you a little flavour of some of the things that I've seen and some of the things in my career from academia, industry and government, which may be relevant to what you're discussing at the meeting. So if I could have the first slide, please. So going back to my time in academia, it was really stimulated by the work of this man, and many of you will know Robert Furchgott, of course, who sadly is no longer with us. But the reason I was stimulated by him was the observation that he made quite late in his career that the blood vessel, the aorta of a rabbit, would only relax to acetylcholine if the endothelial layer was intact. And of course, he described the concept of endothelium-dependent relaxation, and indeed the notion that there was a diffusible factor, EDRF, or endothelium-derived relaxing factor, which later got identified by Gnaro and Furchgott and Moncada and others as nitric oxide. And that observation was an important one that stimulated me because I wanted to understand whether that would also be true in humans. And so my initial research work was to try to look at the responses of blood vessels in vivo in people to acetylcholine, then to experimentally damage the endothelium and to find out what the effect was. Fortunately, around that time, the Moncada lab discovered that the generation of nitric oxide from the endothelium could be blocked by an arginine analog, Ng monomethyl arginine. And that provided a tool to be able to explore this in a much more sophisticated way in clinical experimentation. And so with the ability to have an inhibitor, an enzyme inhibitor, I was able to look at the effects of that on vascular responsiveness in healthy volunteers and indeed in disease states. And a number of things were done. One of them was to discover that actually if you infuse the inhibitor into the brachial artery of a normal human forearm, the blood flow reduces in that forearm. The blood vessels constrict, and that showed that the blood vessels, the arterioles, are in a constant state of active vasodilatation from nitric oxide. And if that's blocked, you see a vasoconstriction. So that was an important pharmacological understanding of some human physiology down to the major discoveries of others that I was able to apply in a human setting. And that ability to do that led me to all sorts of other areas of research. And when I eventually left academia, I looked back at some of the things that I'd done and some of the technologies I'd used. And if I could have the next slide, please. I realized that after many years as an academic, I'd become essentially non-expert in a whole load of things. I was using a range of different techniques to try and answer questions. And you can see the blood flow experiment I talked about in the human forearm at the top of this picture. But as well as those classic techniques, and indeed as well as being always rooted in pharmacology and clinical pharmacology and trying to understand drug effects on mainly human vasculature, but not exclusively that. Of course, the number of techniques that I either used in my own lab or did so through collaboration with others was very much broader and involved cell biology, animal models, of course, molecular studies, structural biology, chemistry, indeed large electronic databases as well from the clinic to try and understand the problem. And I've called this, this was a bench to bed to feedback loop, an iterative cycle of trying to look at things using a range of techniques, both in the laboratory and in the clinic, to try to understand human disease. And I'm not going to go into those studies, but suffice to say that one of the things that I learned, and it was relevant to my own research, was the question is what matters, not simply the technology. So trying not to be bound by a single technology, but to be bound rather by what is the question that you want to answer and which are the colleagues and which are the technologies you need in order to be able to answer that. And that approach to going across technologies certainly was one that I found important. So having spent a number of years as an academic and enjoyed it enormously, and I still, when I'm asked about my career, think that probably the most exciting thing you can do is to be a researcher and discover something, even if it's only for 10 minutes, you're the only person in the world who knows that. It is the most exciting thing. But I became progressively interested in the question of drug discovery. And in many ways, of course, the discipline of pharmacology is rooted in that notion of drugs that ultimately have therapeutic use. And I decided to move from an academic role into industry, and I joined GSK initially as head of drug discovery, and then later as global head of R&D. And I'm certainly not going to try to summarize the things I did there in any detail, but I do want to make just a couple of points. If I could have the next slide, please. The first is that I think there are really four big decisions that one has to make when trying to think about a medicine. The first is, what do you want to target your medicine against? What's your target, your molecular target, or whatever it might be, and what do you want that to do? The second is the medicine itself, and I've called it a molecule on this slide, but of course it needn't be a molecule. It could be a cell, it could be a gene therapy, it could be a cell and gene therapy combined. So the molecule, take it as the medicine, but the question is, what is that? The third is, if you think about the initial clinical experiment, what is it that you want to understand in a human being that tells you that you are on course to be able to make a medicine? And I deliberately called this a clinical biological experiment rather than a phase 1, phase 2 study, because I think that notion of the experiment is central to this. The thing that you really need to understand, it's not a process, it's an experiment that you must try and get the answer to. And the fourth is, if you are on track by that stage, how do you then demonstrate the clinical effect and indeed the value of that clinical effect to healthcare systems? So those are my four things that I think are crucial for the process of drug discovery and development. And of course, they've changed enormously over the past few years. So target identification, I think, has been given a completely crucial new lease of life with genomics and the ability to understand what effect the gene might be having on a particular function in humans. And that is crucial in order to really pin down which target you want to go after. So I think the modern advances in genomics are instrumental in unpicking true target validation. And they're most extreme and most obvious when one is thinking about rare diseases, where you often have a direct causal link between a genomic change and a disease, but increasingly it's becoming very important for common diseases as well. The second one is the molecule that I talked about. It needn't be the molecule. It can be a cell or anything else. But the key point there is it has to be something that has quality, and the quality assurance of which molecule, which medicine, which instrument you're going to take to make your intervention is crucial. And I talked about the clinical biological experiment, the crucial nature of that. And a word about the final one of the demonstration of clinical effect. I think we underplay the value of healthcare systems in understanding this. And if you look across most disease states, the percentage of patients who are in clinical trials who might be eligible to be in a drug trial is far too low. And given that we don't actually know how to treat most diseases, we know, what we know today, but we don't know ultimately how to treat them. And very often we haven't got really strong evidence for what we should do next. It seems to me that the percentage of patients in clinical trials, and indeed healthcare systems across the world, should be much more geared towards running trials, because that's how we get our answer. And rather than it being 1%, 2%, or 3% of eligible patients who are in clinical trials, we should be looking at much, much higher percentages. And we know already that in the best cases, in some examples of pediatrics, where 90% of children with certain cancers are in clinical trials, why aren't we aiming for percentages like that in clinical trials in other diseases? So these are the four areas. And you will, of course, know that this process takes a very long time and very often takes 10 years or more to make a medicine. And that is both a reality and a frustration. But recently I learned that it's not only a frustration, because there is a certain sort of deferred gratification that you can get when you've left industry and you're in another role. You will years later begin to understand whether some of the things that you're involved with turned out to be right, turned out to be important. And last year, when I was in my final year as government chief scientific advisor, two things came through from my time at GSK that actually made my day when I saw it, because suddenly something that you'd worked on and loved and tried to bring through looked as though it was making it. And I'll show you those on the next slide, please. So these are just two examples that came out last year, both of them in the infectious diseases space. On the left-hand side was an example of a new class of antibiotic, topoisomerase inhibitors. And these had been worked on in GSK for a number of years. And the beauty of these was that actually the drug inhibited two different topoisomerases, and therefore quite difficult to get resistance, not impossible, but difficult. And this was a struggle all the way through in GSK. Did the commercial people want to do this, yes or no? Was there a market for this? Did anyone want it? And I was really thrilled last year to see that the new antibiotic gepotidacin actually worked so well that the trial had to be stopped in phase 3 because of overwhelming efficacy. So that was one pleasure. The other pleasure on the right-hand side was one of those stories that you accumulate when you're in industry. And it's the sort of chance discovery, the chance meeting, the chance conversation that led to something. And this particular one was around drugs for HIV. And soon after I joined GSK, the HIV franchise was actually in some difficulty, and a lot of drugs had just been stopped. But the new head of infectious diseases that we appointed, Zi Hong, decided to really push the integrase inhibitors again and have another go, and took 3 molecules through a collaboration with Shionogi into the clinic. And his idea was that he'd take all 3 into the clinic simultaneously and pick the one that had the best properties and pursue that. And that drug happened to be dolutegravir, which is now very widely used. But there was a second drug, which was cabotegravir. And it had a very odd property of a very long half-life. And what you can see on the middle graph there is the plasma concentration against time in weeks. And following a single IM injection, you can see the drug stays around for many, many, many weeks. And this property, which is not what you normally want in a drug, I remember a conversation with a colleague in North Carolina one day, and he said, what about if we actually make this as an injectable drug, and you could have potentially something that could be used for preventing HIV as a prophylaxis, almost like a chemical sort of vaccine approach, or you might be able to turn HIV into a condition where you simply have 4 or 5 treatments a year of an injection, and perhaps you don't need to remember to take a tablet every day. So this again was an idea that wasn't, as you might imagine, universally loved by people on the commercial side of the organization, but it seemed like an important thing to pursue. And on the right-hand side of this graph, you can see this was an extremely effective preventative measure in non-human primates. So it shows the survival rate in those injected with the drug versus those not, and you can see a very, very strong protective effect. So we suddenly have this drug which was long-acting and extremely effective, and I was delighted to see last year, again, this is one of those deferred gratifications, that WHO and others have said this is really, really an important potential tool in the armamentarium for HIV, particularly preventing infections in high-risk populations, which of course largely means sex workers in Africa, but also in other places. So 2 examples of where the 10 or 12 or 15-year timeline that drug discovery and development takes sometimes can play to your advantage because you get a deferred gratification. And these 2 I feel particularly pleased to see come through. So having had experience in academia, clinical medicine, and indeed in industry, I left GSK and took up a post as the UK's government chief scientific advisor. And my thinking behind that was I'd learned a lot in academia. I'd learned an enormous amount in industry, particularly around how teams work, about the importance of quality assured research, the integration of basic discovery with development, with manufacturing, with commercial, with all those things that matter to get a product through. And I thought I could probably apply some of that in a government setting and broaden beyond biomedical research. So I'm going to move a little bit from pharmacology and clinical pharmacology, but I will come back to it because what I'm going to tell you now is relevant to how governments need to think about that as well. So I'll move to the next slide and talk about government. And I want you to do a thought experiment now. I challenge you to think of any area of government, the sorts of things that government have to worry about in terms of policy and operations and the way that they work in society, any area of government where science, technology, or innovation wouldn't add value. And on this slide, I'll give you some examples. Food. Our food supply, clearly highly dependent on how we think about all aspects of science, how we build our cities and towns, how we think about the insulation of our houses, how we think about our energy supply. Of course, healthcare is an obvious one, but also transport or communications with each other. All of these things are dependent upon science and technology. And that's true in everyday science. And indeed, when I moved into government, I spoke to a very senior person from a previous time who said to me, his view, and he was head of the civil service in the UK in the past, his view was that science was good in parts in government and came to the fore at certain times, but it wasn't embedded in government. It wasn't a reflex in government for people to think about science. And he argued that if you go back 50 or 60 years, the same was true of economics. Economics was important, but it was peripheral. If you had an economist, you might ask somebody about economics, but it wasn't embedded. It is now embedded in every government department, and that's true across the world. And the same, he argued, was true for science. It was there, and it could be used at certain times, but it wasn't as fundamentally embedded in the way governments work as it should be. These breadth of examples display why that is a crucial thing to worry about. And if you then move to an emergency situation, and during my first 18 months or so in post, I had a number of emergencies which required science advice. One of them, and there's a picture here, was of a dam that was due to collapse, leading to the possibility of a reservoir emptying right the way across a landscape and causing flooding. Huge number of scientific and engineering questions, not only in terms of the dam itself, but in terms of where that water flowed and what the effect of that would be on sanitation, on power supplies and other areas. We had an emergency one Christmas of drones appearing over a major airport in the UK. And the question that came to me was, well, how do you actually control drones at an airport, given that you can't block the signal because you've got aircraft coming in and out? What are the mechanisms to catch these things or to stop them or to disable them? Something that, of course, requires not only academics, but industry partners in order to do that. And the emergency, which I had just weeks into my job, was of the Novichok poisoning in Salisbury, where 2 individuals were poisoned with Novichok, and the effects that that had, and indeed the environmental consequence of that. So science is important for every day in government, but it's also important particularly during emergencies, and it's important for the economy. If I can have the next slide, please. So this shows a couple of things. First is that if you look across the world and ask which are the biggest and actually the fastest growing companies in the world, science and technology companies comprise 7 out of the top 10 companies globally by market capitalization. If you look at it at a country level and ask what is the relationship between productivity and the R&D spend in that country, both private and public sector, you see there is a positive relationship. In other words, those countries that spend more on R&D, private and public, tend to have better productivity than those that don't. So science and technology is not just an everyday need. It's not just something for emergencies, resilience, defense of the nation. It's also a key driver of the economy. And given that, you would hope that governments are really good at it. That's what you would expect and want, given what I've just said. And I'm going to talk parochially about the UK now, but I can tell you this is true for what I'm going to show you will be true for lots of countries around the world, and is a problem that needs to be addressed. So taking into account the advice I've been given and these observations on the importance of science, I undertook to look at the ability of the UK government to do science. What were the things that made me think I was confident or not? Can I have the next slide, please? And this report was published in 2019. It was called Realising Our Ambition Through Science, and I'm not going to go through the whole details of it, but there are some important things in here that are relevant to other countries and also relevant, I suspect, to some companies, including pharmaceutical companies sometimes. Science budgets have been vulnerable to disproportionate reductions. In other words, it's relatively easy to cut a research budget because you don't feel the consequence until years later. Departments needed a clearer model of science leadership. There was science going on, but it wasn't always obvious how that impacted upon the way a department worked. Resources had grown weak. In other words, people didn't have access to the tools they needed, whether that was specialist tools or whether it was data visualization to help non-specialists understand the consequence of certain scientific aspects. And in the UK, we have public sector research laboratories, as most countries do, and they had tended to get underfunded and in some cases get more neglected than, for example, the universities and other places, and we felt that they needed to be supported. But perhaps the most striking thing was not these big structural changes, but something quite simple, which was if we looked across the graduate intake scheme, so this is the scheme that brings bright young things into the civil service, and those bright young things, of course, will be the future leaders of the civil service, we found that a very, very small number, only 24 or so, were on the science and engineering graduate scheme out of over 1,200 in the whole scheme. And when we looked at the so-called general fast stream, so these are generalists coming into government who might end up in all sorts of different places, only 10 or 11% had a STEM degree. Now, I don't know what the right answer is. It's not 100% should have a STEM degree, and arts, humanities, social sciences are incredibly important, but 50-50 would seem about reasonable. So we found a massive underrepresentation of science coming into the civil service, which is, of course, a problem in terms of the future. So there's work to be done, and indeed, on the next slide, I don't want to bore you on this because it's too parochial for the UK, but we'll just look at it. There were some things that have come through. It's now accepted that science needs to be embedded in government. It's accepted that we need to have at least 50% of the graduate intake needs to have a STEM degree, and it's accepted we need more specialist scientists, technologists, and engineers in government. Perhaps even more important was a document that was published on Global Britain, which had a chapter on science and technology and had science and technology running through each individual chapter. And you look around the world and most governments have got something like this, but very often we don't hear much about science and technology. So trying to get these things embedded in government is important. But government isn't just about, or the science and technology in government isn't just about what happens inside government. It's also about the surrounding things in a country. And I'll just have a couple of slides on this in terms of the UK. Let's start with academia. Next slide, please. You've got to have a good academic base. You've got to have a good fundamental science base. The UK actually historically has had a good science base and actually has had a particularly strong base in biomedical science and indeed in pharmacology and clinical pharmacology. But we can't take that for granted. And although probably the UK punches above its weight in a number of ways, that cannot be guaranteed. And many other countries are very, very good at science and technology now and will continue to get better. So the first message here is any government must fund and protect its fundamental science base and continue to do so, even when times are tough, because it has to be consistent. Next slide, please. This notion of protecting curiosity goes back over 100 years in the UK to somebody called Richard Haldane, who made, I think, two important observations when he produced a report for the government. The first was, and it remains true today, decisions about which research projects to fund should be made through independent evaluation by experts based on the quality and likely impact of their research. In other words, ministers and governments shouldn't decide that, expert panels should. And the other important thing he talked about was a distinction between what he called specific research and general research. And he argued that specific research, for example, the Department for Transport would like to know something about electric vehicles for the UK, that can be commissioned by a department. But general research, which is the sort of thing that many people in this audience will be doing, curiosity-driven research, trying to understand something, needs to be protected separately from specific research. And my final sort of point about the academic side is thinking about new ways of funding. Next slide, please. And the model which in the UK we're looking at now is based on the US model of ARPA, DARPA, and IARPA, and it's called the Advanced Research and Invention Agency. And it's about trying to fund research where you are taking people in different institutions, academic and private, to try and tackle a really big problem together, and using the funding as a way to get leverage across all of those areas and take on really difficult problems. So academia is crucial, government is crucial for science and technology, and so is industry. Next slide, please. And industry is important not only in the big companies that you have in a country, but also in terms of the deep tech startups you have, those companies which are going to be the big companies of the future. I don't want to go on too long about this, but just to show you some trends which I think are revealing. So on the left-hand side, it's the number of venture capital deals in deep tech companies baselined against 100% in 2015, and compares the UK, the US, and the rest of Europe. And you can see roughly the number of deals has increased proportionately. So obviously the starting point's a bit different for those three places, but the number has increased roughly in line. If you look at the right-hand side and ask what the size of the deal is, what you see is the US massively outranks the UK and the rest of Europe for the size of venture deals. And that is an important gap. It's stifling innovation, and it's also part of why we see a problem with scaling up companies in the UK and indeed in Europe and in other parts of the world as well. So a flourishing private sector is the third crucial part of this triumvirate for science and technology that government needs to worry about. Let me move from the sort of process and theory and some of these observations to a specific. And the specific I want to talk about is COVID, which I was heavily involved in as my role as Government Chief Scientific Advisor. Next slide, please. Can I have the next slide, please? Thank you. In January 2020, on the 3rd of January, I was away and I called back to my office and said, I think there's something that we need to really start thinking about in terms of science advice into government, and that is the emerging infection in Wuhan. And if I look at how things evolved following that, the three areas I've talked about, academia and clinical, the private sector and government science, were all important in the response. And I think there are some lessons that come out of that. The fact that quite early on, it was possible to draw on really good experts from a range of sciences, whether that was pharmacology, thinking about therapeutics, or whether it was vaccinology, whether it was mathematical modeling, epidemiology, virology, social sciences. The ability to draw on those people was crucially important for the advice that came into governments around the world, and it was certainly true in the UK. So if you don't have those people in your country, if you don't support a broad science base, you don't have the expertise to turn to when you need it. And that ability to undertake academic research and to pivot to do that on areas of importance for government at the time was really crucial. It was also crucial because in the UK, clinical trials were a big part of what happened in the National Health Service. It was possible to quickly set up a clinical trial to look at interventions for COVID. And I give great credit to the people who led this, Martin Landry and Peter Horby, who set up the recovery study, which was a big study where at one point during the pandemic, and it goes back to my point about the percentage of patients in clinical trials, something like 10 or 11% of every single patient in the UK in hospital with COVID was in a clinical trial. And because of those big numbers and a single large trial, at about 160 days into the pandemic, it was possible to show that dexamethasone reduced mortality and morbidity. A really good example of a clinical trial giving you important, useful information. It was also important because it showed what didn't work. And you'll know that there were lots of people claiming all sorts of things that worked, and anecdotes were becoming the norm. And politicians, of course, like to leap on an anecdote and say, why aren't we doing that? The rigor of doing clinical trials was possible because of the infrastructure and the ability to run trials. So this isn't just important during normal times, it's important during emergencies to have a clinical trial infrastructure working in your country. It was also important that there were people who were doing genomic sequencing, and it was possible to get genomic sequencing of the virus done quickly and see what was changing and where evolution of the virus was occurring and new variants were coming from. So the range of academic disciplines and the application of that was a fundamental, and where I look at where the UK did well and didn't do so well, it did well in terms of the ability of science from academia to come in and make a difference. But what about industry? Well, in terms of vaccines, therapeutics, and diagnostics, the UK didn't really have a significant diagnostics industry. And that meant it was a very difficult time to try and scale diagnostic use with COVID. We have got the big pharmaceutical industry, and that side of things was much better. And we did have access to a large vaccine possibility, and that made a difference. So being able to access industry is crucially important, because this isn't something that can just be done in academia. So the reason I'm laboring this point is when governments think about resilience, and after all, one of the things that governments must do is protect their citizens, they need to think about the industrial base and whether it is sufficient for what they would require in the event of an emergency. Another example from COVID in the UK was we wanted to understand the spread of the virus in the population and to do a weekly, or in fact twice weekly, survey of infection rates from random samples. It was difficult to get the workforce ready to be able to do that, except, of course, we had many people working on clinical trials who were not able to work on clinical trials during COVID, and they became an important workforce to be able to help with the survey that took place, which became widely used across the world as a way of understanding trends in movement of COVID across the population, which parts of the population were most vulnerable. So that ability to set that up was both government science, the Office for National Statistics in this case, and private sector science that provided the workforce from what was a CRO doing work on clinical trials and other areas. We couldn't have done the vaccine program just with academia, even though the brilliant work of the team in Oxford got the what became the AstraZeneca Oxford vaccine, had done all the hard work on that in terms of research. There was much more hard work then to be done in terms of development and manufacturing. That required an industrial base. In this case, AstraZeneca took it on. And of course, the same is true with Pfizer and the production of the messenger RNA vaccines, and indeed Moderna. These ability to scale this is crucial. So you've got to have that industrial base ready. It's got to interact with academia. And government needs to be there too. So none of that works if the regulatory system doesn't tie into that. And one of the things that worked incredibly well was regulators sitting down with academics, sitting down with industry, asking what is coming in terms of both vaccines and therapeutics, and what do we need to do to be at the cutting edge of this. And I remember a discussion I had with the head of the MHRA, the UK drug regulator. I said, would you accept a trial protocol that's been approved by the FDA without somebody having to go through an entirely new process? And she said, yes, we'll do that. And that sort of flexible attitude is crucially important for efficient running, particularly during the pandemic, but you could argue also it could be used at other times as well. So when I look at the overall COVID impact, and of course it was ghastly across the world, horrible time for everybody, it looks to me as though the areas from the scientific aspect, ignoring political and other policy decisions, where we were able to do best was where we had strong academic scientists, where we had an industrial base of some sort that could be rapidly applied to this, and where government also had science that could interact effectively with that. And probably the most obvious example in the UK was the Vaccines Task Force, where we tried to rapidly accelerate the R&D, manufacturing and deployment of vaccines, and as a result managed to get the first vaccines, approved vaccines, into patients. Now, I don't want to stick on the UK. I want to go internationally because this is the World Congress, and I want to show you one more slide on COVID, please. When the G7 was in the UK, I was asked to lead a piece of work which, together with Minister French Gates, on what it would take to accelerate vaccines, therapeutics and diagnostics. And this became the 100-day mission. And we set a very simple target, which is what would need to happen for the world to be able to respond within 100 days of a public health emergency due to a pandemic being declared by the WHO, to have an initial regimen of therapeutics ready for use, vaccines ready to be produced at scale, and a reliable, approved diagnostics. And industry, academia came together to say, yeah, this is possible. Of course, there will be infections that we can't do that for, but we must always strive to get there. And of course, we won't have the tools sitting on the shelf. But we came up with 3 principles, which I think are relevant. The first is the world should restock its armamentarium of R&D products. In other words, make the building blocks, make the technologies to be able to make rapid medicines, vaccines, diagnostics for priority pathogen classes. So whatever the priority pathogen classes are that the WHO say those are the ones we need to be worrying about, we make the initial building blocks to be able to go fast out of the traps in order to be able to make those medicines. And industry, of course, is crucial for that. So there's a whole piece of work going on around that. The second was embed in everyday practice something that you're going to need during an emergency. And that comes back to my clinical trials point. If you're doing clinical trials in everyday practice, you can do it in an emergency. If you're not, you can't. If you're making vaccines or using vaccines routinely in healthcare, you can scale that for an emergency. If you're not, you can't. So this is about working out what countries want and embedding it as everyday practice. Don't, for example, dream that you can set up a vaccine manufacturing facility, not use it for 20 years, and it'll be fantastic when you have the next pandemic. It's only going to be fantastic if it's used routinely to make vaccines during normal times. And the third principle that we said was define the rules of the road and agree them in advance. In other words, don't be trying to negotiate what samples you might want to travel across borders, what finance systems you want, what regulatory systems you want during a pandemic. You know the types of things that are going to be needed. Try to work those out in advance so they click into action quickly when, it's a when rather than if, a pandemic hits. So the 100-day mission remains a goal. It's got work streams really working hard on this now. It's beyond the G7. It clearly is a global thing that requires everybody to be involved. And of course, at the other end of this, there needs to be an equitable distribution of the outputs. And at the front end of this, there needs to be a surveillance system which works to pick up infections quickly. So pharmacology, absolutely central to this, and my view, absolutely central to preparedness for future pandemics. I'm going to end by moving off pharmacology again and talk about one other thing which is crucial for governments and crucial for all of us, which is the next big challenge, which is a durable, long-lasting challenge, and that, of course, is climate. Next slide, please. This slide, which is one of my favorites for this, because actually politicians get this and respond to it, shows the carbon dioxide concentration going back over 800,000 years. And the reason this is possible to do is you can take very deep ice cores from the Antarctic going down a very long way and pick up ice from 800,000 years ago. And in that ice, there are trapped air bubbles that you can measure the carbon dioxide content in. So this is the work of many labs across the world. And what you can see is that the carbon dioxide content bubbles around over 800,000 years until you get to the Industrial Revolution. And the little red line that says today at the right-hand side of this is not an arrow. That is where we are. In other words, it's been below that natural cyclical peak for 800,000 years, and suddenly it goes right off the scale with the Industrial Revolution. So this is a real problem, and the CO2 is linked to temperature. And of course, my three areas of academia, industry, and government are going to be essential for this problem as well. We need the academics to keep working on everything from sub-kilometer scale forecasting through to understanding clouds, through to inventing new technologies. We need industry to build those technologies and to grow them and make them implementable at scale at an appropriate cost. And we need government to be there to support that science and to be part of monitoring the output and ensuring that we get this scaled input. And indeed to do the things that industry can't do on its own, such as fusion and the huge investment that needs to go in over decades to get something like fusion as a viable source of energy. So I won't pursue this because my time is up, but to say this is the big challenge, and this is one that needs those three areas to come together, just as we saw during the pandemic. And my final point, if I may have the last slide, please, is in everything I've spoken about, whether it's how academia works, whether it's how industry works, or whether it's how government works, is totally dependent, in my view, on diversity. So diversity is not a nice to have, it's an essential. You don't solve big problems by everybody thinking the same, everybody coming from the same background, everybody having the same thought process, the same training. That diversity is there to challenge and to bring new concepts and ways of thinking in to improve our ability to deliver. So fundamentally, we need the broadest possible, most diverse workforce, trained differently in order to be able to help us solve these big problems. So diversity, and indeed the thing that follows from that, which is inclusion, to allow this to be truly effective, is a must-have, not a nice-to-have, and fundamental to everything I've talked about. And I apologize once again that I couldn't be with you. I'm sorry you've had me in this sort of disembodied way talking, and I hope that some of the things I've said have at least have stimulated some thoughts as to how the crucial sciences of pharmacology and clinical pharmacology are central to so much of what government and indeed all of us need to think about and hope we get right, because it is the thing that creates the opportunities and the treatments and prevention for the future. Thank you very much for your attention.